all of a sudden we're very quiet. Or do you want to play the theme song?
Good. Be clear if you unmuted, Dave.
It's magic. He's muted, but you can hear him.
People are going to take us as a very serious apartment. We start with playing music.
Well, you know, it just kind of, all right. Looks like we have a good crowd here today. So I'm going to just introduce our speakers today, and we'll, just the order, just so everybody knows. First, it'll be James, and then Anita, and then Dave. If anyone has questions, you can either raise the little blue hand that you all have. You can type something into the chat. You can unmute yourself and ask a question. So you have all those options as far as asking questions. And to start things off, I will actually introduce our panelists. So go ahead. Okay. So our first panelist today is David Block. He's professor and chair of the Department of Viticulture and Enology, and he's also a professor in the Department of Chemical Engineering. We have Andrew Waterhouse, who is a professor in the Department of Viticulture and Enology. He's a chemist, and he's also the director of the Robert Mondavi Institute. We have Anita Oberholster, who's a cooperative extension specialist in Enology, and she's also in the Department of Viticulture and Enology. And we have James Campbell, who is a graduate student in the Waterhouse lab. So James is going to start off today, but I think Andy's going to do a little introduction for James.
Yes, and hopefully James gets reconnected.
Oh, did we lose James?
We lost him there. He was, I don't know what happened to James. Anyway, maybe he'll be back. Hopefully he'll be back shortly. So James is a graduate student in agricultural and environmental chemistry, working in viticulture and enology. So he's really an applied chemist. He's not a winemaking student, but he has a lot of experience in both grape growing and winemaking. And he started his studies at, he's back. He started his studies at Fresno, where he was a student, bachelor's student, finished his degree there and did some research with Jim Kennedy, who I'm sure most of you have heard of. When Jim left Fresno, James Campbell came up to Davis and started his graduate work in agricultural chemistry. And he came with very unusual funding. He came in with research funding from AVF, a project that he pursued his first year. And since then, the project's moved on, and I would say just to summarize, the project started as sort of a survey to try to understand what was happening with tannin extraction, and it revealed some interesting clues about what might be affecting that. And the project has changed, as most research projects do, and now, as you'll see at the end of James' talk, we're trying to address sort of the fundamental chemical questions that are controlling this effect that seems to be altering tannin extraction some way or other. Anyway, so with that introduction, James, I guess you're back online. I was a little nervous there for a second. I was going to have to give your talk.
I just showed my face, and then I just am going to cut off before I actually have to give my presentation.
Okay, your audio is a little raggedy.
I did make you a co-host again, James, so you should be able to share your screen.
Is my audio pretty bad?
You're doing okay at the moment.
Yeah.
Okay. Does that work for everybody? Okay.
Perfect.
So was already introduced, this is the office hours with Dave and Anita, and today we're going to talk about phenolic extractability and reactivity. Briefly, I'll be discussing the impact of maturation and extractability of condensed tannin, along with the possible influence of anthocyanins on the process. I will also be discussing how anthocyanins seem to readily be incorporating into condensed tannin and modifying it through decreases in polymer size, and that's some of the kinetics work I'll be doing this year, hopefully, if I can ever get back in the lab. And also some of the previous work done on sensory stuff. So just an introduction to polyphenol synthesis. Generally, we're discussing polyphenolic synthesis in the developing grape as bimodal or asynchronous from flowering to the onset of aeration is where condensed tannin is produced in grape skins. However, after lag phase, you basically see a stop to the production of condensed tannin, and then there's a shift in the phenylpropanoid pathway to the production of anthocyanins, so condensed tannin is no longer produced. So basically from the onset of aeration to commercial harvest, all of your tannin that was produced, it's done, and there's just modifications to the tannin happening during that time, or we think there are. So as far as tannin extractability is concerned, I did 5 time points in this project, but really anything before 22 or 23 bricks doesn't really matter to winemakers, so I only am showing these for extraction. This is a project I did in 2018 with Sauvignon Blanc. Basically, I did skin extractions with 0 milligrams per liter of isolated anthocyanins added, then 300 milligrams per liter and 1,000. And what we saw was that it didn't appear like there was any changes in tannin extractability from 22 bricks to 24.5 bricks. And Anita will probably talk about some of the absorption, desorption work that she's done with her group later. But it didn't seem like there was any changes in extractability as far as this maturation time. And I think I might get a question about why I didn't go further later, but for right now, I just wanted to show that. Well, what I really started this project to understand, though, was what about anthocyanins? So when you add anthocyanins, is it enhancing or not enhancing at all tannin extractability? And from this graph, you can see that the first 3 time points, it seems like there's a difference in extractability between the control and the treatments. However, at commercial harvest, or 24 bricks, 24.5 bricks, there is no actual significant difference between having anthocyanins in the system or adding anthocyanins. And also at every single time point, there was no significant difference between the concentration of anthocyanins, so 300 milligrams per liter or 1,000, which I found interesting. So it suggests that there might not be an enhancement, even though previous research has suggested that there is indeed an enhancement in anthocyanin concentration on tannin extraction. Now, the second part of this is going to just talk about some of the other work that I'm interested in as far as tannin modification. Before I get into that, I probably should discuss some of the implications of tannin that from a winemaking or wine drinking side. Generally, some of the sensory studies have discussed tannin as far as molecular mass or size as increasing astringency. So really large skin tannins that are extracted during the winemaking process can be quite aggressive, or I guess chalky, depending on the term you want to use. This was done by Stefan Vidal in 2003, and then Sun et al. In 2013. Also, can you guys? It says unstable. Can you guys still hear me pretty well? Okay.
Yes,
also in 2004, Stefan Vidal did some work showing that pigmented tannin possibly decreases or lessens the astringency of that polymer. So one of the things that I've been interested in since the 2015 survey or project with Jim was what happens to or modifies that polymer to decrease the astringency is is color incorporation basically decreasing the size of the polymer. And what we saw with the 2015 work, this is a PCA from about 365 berry samples in the Napa Valley. And one of the things we saw that was there's an inverse relationship between the monomeric anthocyanins present in the skins and the molecular mass of the tannin extracted. And we used that, or I used that information after I left Fresno State when I came to Andy's lab to basically develop projects to better understand this relationship. So this is some results from a project on Mount Veeder with the Thomas Sheet Estate in conjunction with Will Thomas. Basically, we did a shade cloth trial, and what we're interested in is if we could possibly modify the amount synthesized, the anthocyanins, and how that would impact the tannin that was extracted. And what we saw was that if I plotted the size of the tannin extracted versus the concentration of monomeric anthocyanins extracted, there's a very good relationship between the two. The R-squared on this linear regression is 0.78. However, it's not going to be linear. I put a linear regression on here. It wouldn't be linear. It'd be more like hyperbolic, because at some point you just form dimers. So it's not a true linear relationship. So in 2018, back to the Sauvignon Blanc project, one of the things that we were really interested in with gel permeation chromatography is understanding if I add various amounts of anthocyanins in a system that had no anthocyanins to begin with, what would that tannin size look like, or what would happen to that polymer? And what we saw was a very clear, Dr. Waterhouse and I saw a very clear treatment effect, wherein as you added more and more anthocyanins, you decrease the polymer size of that tannin. So or it appears as though you do at least, but from the earlier sensory work, it could be possible that the reason why we're seeing variations in astringency when you have pigmentation is due to variations in polymer size happening. However, there is some new research suggesting that anthocyanins themselves are astringent, so we're not really sure yet. So that was very fast, lightning round, I guess. But the conclusions are that as far as extractability percentage of skin tannin, it doesn't appear from my work that it's significantly changed in later stages of ripening. The addition of anthocyanins doesn't appear to have a treatment effect on the extractability of skin tannin. But I know that other researchers have seen different. However, we use different methods of quantification in comparison to those other studies. The presence of a nucleophile or a terminal subunit, such as anthocyanin, can decrease the molecular mass of the condensed tannin polymer. So it's decreasing the size of the polymer itself. And the next project that we're really interested in is that anthocyanins appear to incorporate into condensed tannin readily. The earlier work from 1983 suggested that the kinetics for these incorporation reactions are really slow on the scale of weeks or months, and all of these extractions happen in 72 hours. So it happened relatively fast, and we're thinking that we need to reevaluate those kinetics. So as far as acknowledgements, I'd like to acknowledge Dr. Waterhouse and the entire Waterhouse lab, Pat, T, Brian, Dr. Kennedy, Florian Gronickel from the University of Dijon, who was my intern last year. Now he's at IOVV. Maury Anderson, who used to be the lab manager, Hush Vineyards, Constellation Brands, Thomas Shea Estates, and all the funding agencies, including my fellowship scholarships and the American Vineyard Foundation. Was that fast enough?
That was pretty
Sorry,
good. Thank you. Now, if you want to unshare, Anita will
stop
share.
share.
Yep.
Maybe I went too fast.
What does that look like? Is that correct?
Yep.
Okay. Okay. So I'm going to go pretty fast. I hope you follow the accent. So to continue, so we've seen that phenolics are really important to red wine quality. So there's been plenty of winemaking techniques that has been developed to mostly enhance the extraction of most phenolic compounds. So what I'm showing you here is all the parameters that the Bloch and Oberholster lab have been looking at since 2011. And I'm just going to give you the main findings from each study. So this is going to go pretty quick. So firstly, we looked at temperature and extraction profiles during red wine fermentation. And we did this by using our 2,000 liter or 500 gallon tanks. And we had this curtain of temperature probes, and we also have sampling probes at different levels. So if you look at the next slide, you'll see here, we have contour plots that we constructed from the ports that you can see at each of these levels, these color bands. We had 3 ports at one level, and we had 4 different levels. So 12 sampling points gave you this contour plot. And you see that's for 6 days. And what you can see there, you see the extraction of anthocyanins, you see the buildup of anthocyanins in the cap and the liquid portion just below the cap compared to the liquid below. So all these samples were taken just prior to a pump over. So you could see the buildup that happened. And you can see after about 4 days, 96 hours, very little extraction from the cap happened, which is pretty, agrees a lot with other research showing that anthocyanins extract really quickly. And after that, you don't really get much extraction anymore. The graph here on the left is just showing you the average of the 3 ports at one level. And it shows you that after 4 days, you basically, your mass concentration and your cap concentration is the same. In the next slide, we also, for a few days, we took hourly samples. So the first sample is just before pump over, and the second sample that you see is just after pump over. So first, it proves how good the pump over worked, right? This was 1 volume pump over twice a day at 9 a.m. and 5 p.m. At 4 p.m., that's the hour just before pump over. You can see you actually have the gradient forming again. This was at day 3 of fermentation. Day 5, we didn't see this buildup because there was no active extraction from anthocyanins from the skins anymore. When we did the same thing for tannins, I'm not showing you the data, but then we just saw an continual increase because we know tannins can extract for weeks and weeks, unlike anthocyanins that extract pretty quickly. I always forget my animation. Next, we looked at the impact of cap versus liquid temperature. We did this using our research fermenters. There are about 50 gallons. We ferment about 30 gallons in them. We use our integrated fermentation control systems, and basically attached to that, we have a jacket temperature probe, a cap temperature probe, and a mass temperature probe. We ferment it at 25, 30, and 35 degrees. For one set of treatments, we kept the cap and the mass at the same temperature. So basically, we said if there's more than a degree variable between difference between the cap and the mass, do a pump over until it's the same again. For those treatments where we wanted a different temperature between the cap, a variable temperature between the cap and the mass, we said, okay, 5 degrees. We found that we can easily have 5 degrees in these fermenters. We'll say if the difference is more than 5 degrees, only then do a pump over. And what we found was that differential was only really there for the first 3 days of fermentation, of active fermentation, because after there just wasn't enough heat build up during fermentation. And I show you only one data slide from this. And I'm only showing you the treatments where we kept the cap and the mass the same, because we found that that differential really didn't have a major impact. The driver for extraction was the mass temperature, because it's only 3 days that you have the differential. And you'll see increasing the temperature, increase in catechin extraction. We saw the same for a lot of other markers and for tannins. Catechin is an indicator for C tannin extraction. So we did see more C tannin extraction compared to skin tannin for higher temperatures. And for molybdenum-free glucoside as an indicator of all anthocyanin or color extraction, we saw that although it extracted faster at higher temperatures, you always ended up with the same amount because they extract so easily. Except for at 35 degrees, we saw a significant decrease in color by the end of fermentation, which we could not explain by the formation of polymeric pigments. We think we actually saw color degradation when the cap temperature went 35 degrees and above. So next people said, yeah, but you know, you did a lot of different pump overs trying to maintain these temperatures or temperature differentials. So then we looked at the impact of pump over for 2 seasons. We did 4 volumes in total, doing 1 time pump over twice, 4 times, or 8 times a day. And we saw no impact, no matter what compound we looked at. But then 4 volumes is quite a lot for such a small fermenter. So we did go more extreme, where we looked at half a volume twice a day, half a volume once a day, quarter of a volume twice a day, and no pump over. And we saw basically exactly the same graphs than you see here, except for the no pump over, there was a slight decrease in extraction in some compounds. Now we'll say, yeah, no pump over, no impact. The fact is we had pretty small fermenters. So you do get natural mixing due to temperature gradients forming, as well as the release of carbon dioxide during fermentation. So in a commercial scale fermenter, this may make a difference, obviously. So next we looked at cold soak. Now, what's the mechanism of cold soak? The idea behind cold soak is you have an aqueous environment and you are promoting the extraction of hydrophilic compounds like your color compounds. And you're also enhancing extraction from the skins because you have no cap forming and you get no more breakdown of the skin structure. However, this doesn't gel that well with what we know scientifically about extraction. We know that both anthocyanin and tannin solubility increases with higher temperature and higher ethanol content. And we know tannin can actually also extract in an aqueous environment. It takes a few days for the seed coat to hydrate, but after that, they extract pretty well. So what we did is we did for 2 years, 2 different Cabernet Sauvignons with different phenolic contents. We did a control, 1, 2, 4, 7, and 10 days cold soak. And for 1, 2, and 7 days, no, 2, 4, and 7 days, we also 1 year did 30,000 gallon commercial comparison, and they found the same results, basically, the same trends. Even though at the start of fermentation, you had different amounts of color or molybdenum-3 glucoside, depending on the length of cold soak, by the end of fermentation, which for us was only 7 days, they were all the same. Because color extracts so quickly, if you have a hot fermentation, by that I mean jacket temperature of 25, by the end of 7 days, they're all caught up. We did see differences or trends in differences in skin and seed tannin. The longer the cold soak period, the more tannin you started off with, and those differences were maintained because they continue to extract. We also saw at 7 to 10 days, you started to get a trend of significantly more seed tannin. So that is definitely something to look at if you're going to do extended cold soak periods, because you can start getting more coarser and bitter tannins. So what we basically learned, you do you do pump overs because you're trying, and other cap management techniques to promote extraction and not have saturation, to control the temperature, because like we said, above 35 degrees or 95 degrees Fahrenheit doesn't seem a good idea for color extraction, and also to keep your yeast happy. Above 42 degrees C, you will start inhibiting your yeast. It seems like a 1 to 2 volume pump over, depending on your fermenter, will do a pretty good job at removing any temperature or concentration gradients in your fermenter. And that complex pump over techniques probably isn't as essential as some may think. But now, to really understand the mechanism of extraction, we really need to understand the mechanism of extractions to allow us to better manipulate phenolic profiles. So after we had all this data, to really understand what we saw, we started doing more basic research in the lab. And I'm not going to go into the details, but basically what we focus on at this point in time is on the release part, right? But we also know after phenolics get released from the cells, they can take part in different reactions. One is tertiary reactions forming polymeric pigments, for example. Others is actually readsorption onto solids, onto yeast, onto cell wall material. And then you can also get desorption. So all of these are in equilibrium, and we think they're definitely a function of temperature and ethanol. So this is what we investigated. And basically what we did is we isolated anthocyanins, we isolated cell wall material, we added them at different concentrations at different conditions, different temperature and different ethanol conditions. So what you saw there, we did 15, 22.5, and 30 degrees C, and we did 0, 7.5, and 15% alcohol. And we did exactly the same thing for seed and skin tannin at different concentration, and then combinations of. What we found was that you have less absorption back on the cell material at higher concentrations of ethanol and at higher temperatures. It makes sense because then they're more soluble in the liquid phase. We also saw that all absorption is reversible. And desorption happens faster the higher the temperature, the higher the alcohol concentration. And this is why you can do donations of skins and many times see desorption again happening. The other thing is that we also noted that there's a definitely a trend of higher or larger tannins preferentially binding because they're bigger and they can bind to more binding sites on the cell material, meaning skin tannins that's larger than C tannins will preferentially bind. If you combine anthocyanins and tannins, they compete for binding sites, and you will get less anthocyanins binding and more tannin binding. So those are the combinations that we saw so far. And basically, all this data was used by Dave's lab to do mathematical models to predict under different grape composition and ethanol and temperature conditions, what kind of phenolic extraction we'll see. So that is all for now. There's lots more work I can share with you, but for that we need a day. Acknowledgements, lots of people have been working on this project since 2011, and obviously funding sources, of which the most important is probably E&J Gallo Winery for their support for all the years. And with that, I'm passing the ball to Dave. As soon as I stop sharing my screen.
Exactly.
I knew you were going to say that. All right, before Dave starts.
Thank you, Anita. That's nice of all of you to show up for my birthday today. Thank you. All right, so what I want to do in about 4 or 5 minutes is to kind of continue on with where our research went after what Anita presented. So I'm going to share my screen and do that now. All right, can you see that? Okay, good. So what I'm going to talk about is developing a model, mathematical model for trying to predict and understand phenolic extraction. There are really 2 reasons why we're doing this. One is, as Anita just mentioned, there are things that we found in our experimentation in the winery that we couldn't explain, or at least not very well. Things like, if we didn't pump over, why do we get the same amount of extraction? And sometimes that happens at a larger scale and sometimes it doesn't. You know, why does it look like the liquid temperature might be more important than the cap temperature? These are all things that we wanted to be able to understand better, to be able to use that information to optimize phenolic profile, basically to get the phenolic profile that you're shooting for as a winemaker, whatever that is. And the second reason to do the modeling is to be, if you have a good mathematical model, you can actually predict what's going to happen from your initial conditions and what you're going to do with processing. And if you can predict what's going to happen, it means you can optimize what's going to happen. You can use it as a tool to get closer to your goal wine than you might otherwise. All right, so I'm going to just very briefly go through the model. So the first thing we wanted to do is to come up with a model, a 3-dimensional model, predicting temperature and ethanol concentration as a function of where you are in the red wine fermenter and over time. Because we know that all these phenomena that Anita talked about and James talked about are temperature dependent, and they're a function of ethanol as well. And so in order to predict phenolic extraction, we need to be able to predict temperature profiles in red wine fermenter, and we need to be able to figure out where the ethanol is in the fermenter over time. So it's not necessarily easy because red wine fermenters are not homogeneous. You have the cap at the top and the liquid in the bottom and so forth. So the model to predict temperature and ethanol consisted of a couple different parts that I'm showing here. So the first part is a yeast growth model. So this is a model that we published probably close to 20 years ago now. This just accounts for yeast growth that's nitrogen limited, and it accounts for the production of ethanol. And then we need all the physics. So we need the first equation here is the mass transfer. The second equation here is fluid flow, and with a semi-compressible fluid. The third equation is the heat transfer. So we need to know all that. And we need to know the physical properties that weren't necessarily measured. So I should mention this is all work of a student of mine, Conrad Miller, who just finished his PhD in chemical engineering. Turns out there was no information on thermal conductivity of grape pomace, so he went out and measured it. And that's what this is over here in the left-hand corner. All right. So with all those things, we could actually go through and predict temperature profiles in a fermenter. If you saw this on Facebook, you saw some of the predictions of that. They actually match experiment really well, which is great. I'm not going to go into that today. All right. So once we have that model, which we do, and we've proved that it works really well, then the next step is to have a mathematical model for phenolic extraction. So this is that same figure that Anita showed with the release of phenolics, the reaction of phenolics, and adsorption, desorption. And again, all of these things are going to be a function of temperature and ethanol concentration. And given the work that we've done together, we know what that function is for the most part. And so we can actually model that. And once we have that modeled, that means that with these two parts of the model together, we should be able to predict phenolic extraction in a red wine fermenter fairly well, regardless of scale and shape of the fermenter and so forth, and processing. So this is just a picture of the model, the mathematical model for the phenolic extraction part. That's the left side of that box, that yellow box they had in the previous screen. The details aren't all that important, other than this is a mass balance. These equations are the release. This is the release step. This is the reaction step, and this is adsorption and desorption. And with our data, we were able to calculate all those parameters. So this is the model prediction on the right. And this is the experiment on the left. So Anita didn't show the tannin, she showed the anthocyanins. So here on the left, that's the other part of that data set that where we measured these things experimentally at the 500-gallon scale. And these are the tannin profiles that you see over time. And here you see for these two curves are in the liquid, these two curves are in the cap. And this is the prediction that Conrad did with that comprehensive model. And you can see it's not perfect, but it's really good. And it tends to explain, in all the other work that we looked at when we made these predictions, it tends to explain what's going on experimentally really well. So that's great. That means we have a model that can predict phenolic extraction as a function of everything from the nutrient levels to the shape of the tank and the size of the tank and so forth. All right, so what do we use this model for? Now, we can use it to understand what's going on, and we've done a lot of that to try and understand, why can we get good extraction in some tanks if you don't have pump overs and so forth. But the other thing that we want to be able to do is to use this model to optimize our processing. And I like to think of this as kind of a GPS system for your fermentation. So the idea here, I'm just showing a screenshot of my GPS on my phone. What you like to be able to do is put in your grape characteristics when your grapes come into the winery, whatever those grape characteristics are that are important. And then your destination is your final phenolic profile, and have it suggest to you what's the best route of getting there. And there may be more than one routes like there are in your GPS, but what's the best route of getting there? And that's what the model can do, is it can help you predict what's the best way of getting to the phenolic profile that you'd like. So just as a quick case study, and this is my last slide that I have, what if, for instance, you wanted to maximize the tannin coming from the skin? And Conrad did the simulation, and what he found was that for a 2,000-gallon tank, he found, for instance, that 1 pump over per day gave more skin tannin than either no pump overs or 8 pump overs a day. All right, so somewhere in the middle is where he got the most skin tannin predicted. And I don't think that's obvious that that would actually happen. And this is the kind of thing that you can use this GPS kind of system for, if it were fully developed, to be able to optimize your phenolic profile for whatever you're shooting for. So that's just kind of an overview. This is a summary of what I already said, but I don't think I need to go over the summary because I want to leave time for questions. And so I'd like to acknowledge American Vineyard Foundation that funded this work. Conrad Miller, the first name there, is the one that did almost all the work that I just showed. A lot of collaborators, including Ian J. Gallo, for some of the funding for this as well. So with that, I'm going to stop sharing and pass it back to Karen.
All right. So this is the time where if you guys have any questions, feel free to ask. And maybe if there aren't any questions coming forward, I will start out with one for Anita, perhaps. What does the no impact of pump over really mean on a commercial scale?
Like I mentioned, I would definitely not recommend that on commercial scale you don't do a pump over, because we do realize, like I said, it seems that it's pretty important to try and keep below 35 degrees C or 95 degrees Fahrenheit. But I think it does mean that you can do some investigations to see, do you need 1 or 2 volume pump overs to get rid of your temperature gradient and your concentration gradient, or do you see impact with normal spectrophotometry? You can work that out. And it seems like twice a day is a pretty good idea, because we've seen a clear buildup during the course of the day. And we've even seen cases that, people couldn't do pump overs because the electricity was shut down. You can actually make up for no pump overs by doing pump overs later, and then actually get rid of those concentration gradients and get the extraction that you need by doing a few days of extended maceration. Obviously, the one concern is if you lost color, you can't get it back. And if the temperature in the cap went high enough that you actually started inactivating your yeast. And then you would know it because you would have a stuck fermentation.
Can I add to that?
Sure.
Since it's my birthday, I can do that, right?
You can always do.
Yeah. So I think I would add a couple of things to that. First of all, a little caveat here on talking about pump overs. Pump overs are going to affect more than just phenolic extraction. We didn't talk about that here. So, the temperature is going to affect the yeast kinetics, how fast the fermentation is going to finish, which could have implications on other things like aromas and so forth that stay in the wine. We're really just focusing on phenolics here. And the other thing that I would say is what we found from the modeling is that one of the important parameters on whether this no pump over thing will work is how far it is from the bottom of the tank to the bottom of the cap. And so basically the taller and skinnier your fermenter, the less likely a no pump over is going to give you the same results as a pump over. But if you had a short, wide tank, and so for instance, let's say, I don't know, little tiny short squat tank or a picking bin or something like that, you might be able to get away with no pump over and have the same extraction. But again, there might be other implications. You probably want to keep the top of your cap wet and so forth and avoid having acetic acid buildup and so forth. So there are other implications. But in terms of phenolic extraction, the shorter and wider the tank, the less likely you're going to need a lot of pump overs.
So Clark, do you want to ask this question, or would you like me to ask it? Solubility of anthocyanins is increased by alcohol, but don't you think the effect is far outweighed by the instability ethanol causes to copigmentation colloids due to its effect on the dielectric constant, the driving force of colloid stability, ?
So we did look at copigmentation as well in these tests. The impact of alcohol on absorption was actually greater than the copigmentation impact. So what happened with copigmentation is, with an increase in alcohol up to a point, it decreased copigmentation, but at 15%, it's like a tipping point, it actually starts increasing copigmentation again. And what we found was, why it wasn't that simple was that copigmented stacks, right? When copigmentation is basically anthocyanins, if it's anthocyanin interactions, it's anthocyanins, and because we had nothing else in our little model systems, basically these anthocyanin stacks could attach to cell membrane just like that as a stack. And that's another reason why we didn't see as great an impact of copigmentation as you would expect, because we had basically these contrasting effects happening at the same time.
All right, I have a question for James. If there isn't another one, I don't see one in the chat. I don't see any hands raised. Most wineries are harvesting red varieties around or greater than, let's say, 24.5 Brix. Why did you stop sampling when you did? And what would you expect to happen with extractability after that?
Can you hear me? It's having problems with my speaker. Okay. First off, I was using Sauvignon Blanc in Mendocino County, and if it went past 24.5 bricks, it was going to have a bunch of botrytis. So that's the first thing. The second reason, well, as far as extractability is concerned, I know that and Anita might be able to say something to it, but I would actually expect it to possibly go down. If you look at Karen Benden's work in 2012, where she used the scanning electron microscope, you actually start to see the cell wall material become more porous. And as it becomes more porous, you start to have an increase in surface area. So you have a wider area for cell wall interaction, tannin interactions. So there's a possibility that extractability actually goes down in that point in time. But as far as why I, didn't look at anything past 24.5 bricks, it was mainly due to disease pressure.
So I can add on to that. You have two competing interactions happening once again. You do get what Karen said, that you have more basically sites for reabsorption to take place, but you also get more extraction or release because of the breakdown of the cell wall membranes itself. So you once again have these two competing reactions taking place.
I'm just pausing to see if anyone has any particular questions, but if I don't see one here, I'll ask one more. And this is for Dave. What are the key steps to commercialization of this technology?
So, so I think, where we are right now, and that thinking about the GPS kind of thing, is if you actually took your phone out of your pocket and put in a destination and had to wait there for about 10 days for it to tell you how to go, you would just not use it, right? So each time we simulate a fermentation, and when I say we, I mean Conrad simulated a fermentation, it took about a day. And so if you're going to iteratively try and come up with the best conditions, it's not going to work with the speed that we're at now. So one of the things we have to do is speed up the computational part dramatically. And so we've started working on that. And that the way we do that is try to capture the important knowledge from that multi-physics model in a neural network. Because once we train a neural network, it'll be fractions of a second to do the whole optimization, as opposed to days and days and days, the way if we use the software that we're using now. So we're kind of far along the way doing that. Not quite there yet, but we have a lot of past experience with artificial intelligence and so forth. So we're doing that to build the model. The second thing we need is to know the total phenolics in the grapes at the beginning. And we need to know, so James talked a lot about extractability. A lot of the assays for extractability are fairly slow. And so what we need is an extractability assay that would take on the order of, let's say, an hour, as opposed to hours or days, right? And so in some work that we did with Anita and Christina in her lab, one of the postdocs in her lab, along with Conrad, we have figured out some parameters that we think are highly correlated with extractability. And we're still testing that and working on it. But if something like that works, I think it would take a little bit to develop an assay for those things that we measured, because our assays right now are still pretty slow. They're not an hour or less. But if we can develop an assay for those things that we measured that are an hour, if you had an hour to measure total phenolics and extractability, and you had the GPS type system that worked really quickly in seconds, then you could start using this commercially. And it's, I think we did put in a proposal to ABF this year to do that, but it wasn't funded. So hopefully at some point we'll still do it. We're kind of working on it slowly in the background now, and we'll see what happens. But I do think that it's not that far off if someone were to work on it full time.
All right, we have a couple of questions in the chat. The first one is, does the cell wall material in the tank mimic the cation exchange complex in soil? As a metaphor, obviously not the same scientific. Who wants to take that one?
I'm going to let a chemist take that one. Maybe we can talk a little bit about what we found with the adsorption of both
Yes.
anthocyanins.
So it's actually a combination of interactions. There's some ionic interaction, but it's mostly coulombic interactions, or in those of us that are a little bit older, when we did chemistry, what we used to call dipole interactions, and some hydrophobic interactions. What we really found was that it really was more about binding sites available than whether the composition of the cell wall material itself and whether it would be preferential to one of those type of interactions. We just saw more binding, the cleaner the cell wall was, less binding, the less clean it was, and less binding sites was already taken up. So it was more a spatial issue, more of three-dimensional modeling issue than it was really what interactions could preferentially take place. Does that answer the question?
I'll let the person who answered maybe say something in the chat. Meanwhile,
Okay.
another one from the chat. Do we know at what point, the person said yes, answered the question. Do we know
Good
at
job,
what
Anita.
point in extended maceration will colors start to desorb?
So the thing is, during extended maceration, you're not really, well, your temperature is constant and your alcohol is what your alcohol is. So you're gonna have an equilibrium. We only saw desorption because we actually removed the saturated liquid and gave it new clean liquid. So there's once again a concentration gradient and it reached equilibrium. So no, you're not gonna start with longer time period without not adding new juice or changing the equilibrium. You're not gonna see more extraction of color. The changes you're seeing is because more of your monomer may be changing, maybe interacting in copigmentation reactions, and may start actually polymerizing, which will change the color that you see, but it's still the same actual molecular amount of anthocyanins in the liquid.
And this one's actually for James. Why is there such a large difference in tannin extractability between treatment and control at 15.5 Brix?
So my answer for this is probably going back to some of Singleton's work in the early 90s. He did some work with, I think it wasn't Sauvignon Blanc, but it was another white variety, I believe, and he used anthocyanins extracted from Limburger. And what he saw was that the tannin wasn't very soluble that was extracted in the white varieties if it didn't have any anthocyanin incorporation. So you lost solubility and you had precipitation of tannin actually in the white ferments. And I think that in some of my cases, I had possibly some of that happen. So at the 15.5 degrees Brix, I mean, it just seems odd that it's so much lower than everything else in comparison to the 300 and 1,000 milligram per liter. But we don't see any difference in the 300 and 1,000 milligram per liter. Am I muted?
No, we can hear you.
Oh, I just got Scott McLeod said James is mute.
No.
Anyways, yeah, so I think that it has to do with possibly precipitation of tannin. That's it.
Before I ask the next question, I am going to announce next week's office hours topic, and that is the impact of smoke exposure on grapes and wine. And we'll have as our office hour panelists, we'll have Elizabeth Tomasino from Oregon State University, we'll have Tom Collins from Washington State University, and we'll have Anita Oberholster, again, from UC Davis. They've been working on a project together and also individually in their own states, and they'll have a lot of information for everyone. There was a question here. What about tannin absorption during extended maceration? Can we expect it to increase, but also change in how it's bound?
So tannin absorption, okay, so the absorption happens actually pretty quickly. You get equilibrium pretty quickly. So that's gonna happen, whatever extracts, you're gonna have release and absorption happening. And it's true that you do get more absorption the higher the concentration in your liquid. So as it is as you get released, you'll also get a certain percentage of absorption. It really varied between 30 to 45%. And basically, in a red wine fermentation, because you have anthocyanins as well, if they're reacting with the tannin, basically you're removing that from the equilibrium. And this is why many times with anthocyanins present, you see more tannin extracting, because it's a new molecule that's forming. So that allows the release of more tannins. So you're going to the whole time you're going to go and reestablish the equilibrium between release and absorption. When that is equal amounts, that's when you're going to stop seeing extraction happening. But obviously there's a limit to how much can absorb, because I just said it depends on how many sites there are for the tannins to attach to. So when that is complete, you're going to start just seeing release. But we know tannin extraction, depending on what's in the grape still has the biggest impact on what you see, can continue to extract for up to 45 days.
Let's see, there's another one here in the chat. Hello, have you done any research on extraction of resveratrol and the effect of ethanol and temperature?
I have not. We have not looked at resveratrol. The problem with resveratrol is normally in most grapes, the amount of resveratrol present is really, really low. So if you just do your normal phenolic profiling methods, you actually don't always see resveratrol. You need to do a separate technique that really focuses on resveratrol itself, and you need to clean up your matrix to be able to see it. It probably, I would think that you could increase in extraction with temperature and ethanol, but they also can be unstable. It depends on what form they are. Most of them are glycosylated in the grapes and can also actually change into different isomers. So, you know, if you look at resveratrol, many times we basically focus only on resveratrol because you need specialized techniques to analyze them fully.
How does pressing affect phenolics?
It's basically physical force. So when you press, you do see a jump in the phenolic content. If you take a sample in the wine prior to pressing and after pressing, you see a 15 to 30% increase because you're basically forcing some of the things that's on the skins and still in the cells out during pressing itself. I don't know whether you're forcing things that was adsorbed to desorb or whether you're just pressing things that was actually still in the cells out. I'm not 100% sure.
Or alternatively, I suppose it could be that when you press as you release those other phenolics, they don't have the opportunity to reabsorb onto the skins, which might make a difference as well, right?
Yeah, that too.
Are there any other questions out there?
Otherwise, can I ask a question for Dave?
You may.
So, Dave, so I know you did this, but I thought it'd be interesting if you tell people that you did this. So you did with the modeling, you looked at the temperature gradients in different size and dimensions of fermenters. Mm-hmm. And you guys also did this on egg fermenters, and it would be perhaps cool if you could just give a summary of what you guys found.
So yeah, I mean, what we found with regular kind of cylindrical fermenters is at some point, as you get larger and larger, your surface area, your cooling area per volume goes, as you get larger, your surface area per volume goes down. And so it becomes more and more difficult to control temperature in a large tank. And I think people who have done processing on a really large scale understand that, which is why at some point you just can't use a jacket anymore and you have to take the wine out and send it through an external heat exchanger every so often. But because we could actually predict temperatures, and we did, and this was kind of a fun side project, sitting there with Conrad, my student, saying, well, if you could predict temperatures in a fermenter this well, do you think you could do it in a concrete egg? Because you know, in talking to people who sell concrete eggs, many times the two things that they talk about are that your temperature control is better because your concrete is a heat sink, and because of the shape, you get better mixing. And actually, quite honestly, neither one of those things is very straightforward from an engineering point of view. It's not obvious that that would be the case. So he did do some modeling, and this is probably a year and a half ago or so. Did some modeling in concrete tank. He actually went and did experiments at Cade out in Napa, where they had some eggs. Did a bunch of measurements and so forth. Found out the model worked pretty well on that too. Not only these are for white fermentations, so not only the fermentation inside, but the concrete outside. You can model the temperature gradient through that. And basically what he found was that concrete acts better as an insulator than it does as a heat sink. And so if you put white wine in a concrete tank, it's going to heat up. That's why a lot of concrete tanks now are sold with separate, you know, heating fins or with cooling coils built into the concrete. And the other thing that he found was if you have a jacketed tank, because of the temperature difference between the jacket and the liquid, you end up getting better mixing in the same size cylindrical tank than you do in an egg with the egg shape. The velocities are higher in the cylindrical tank with the jacket. So it's kind of an interesting side project that we published probably about a year or so ago in the Australian Journal. So it's great when you have a really good student who can sit there, listen to your ponderings, and then, you know, a couple weeks later have results that tell you whether you're right or not.
Anyone else have any questions? Got a few more minutes. If anyone's questions.
Thanks for all the happy birthday messages. I appreciate that. That's great.
Looks like we have one more here in the chat. What is your advice for a situation for unripe tannin extraction control in situations where sugar accumulation and potential alcohol have outstripped phenolic ripening? Would it be preferable to pick earlier with less ripe tannins but less soluble tannins?
Anita?
I'm trying to think about.
I mean, I guess the question, my first question would be, what do you mean ripe versus unripe tannins, or what, like phenolic ripeness? What do you mean by that? And that would be where I would start from. So I kind of need some context. Anita, you know what I mean?
Yeah, I didn't understand the whole question, but I would say if you have unripe tannin, and you know, many times what I do is I look at the seeds, and the seeds green and taste horrible, then the skin tannin, many times even unripe, isn't that bad. So I would focus on getting the color and the skin tannin out, but try and limit seed tannin extraction as much as you can. It basically means not too hot fermentation, keep it between 25 and 30 degrees. So that is 85 and 90. And then, you know, when you have your skin tannin out, I wouldn't do massive amount of extended maturation and things like that, because you're probably going to get a lot of bitterness and greenness. And obviously, if it's unripe, you don't want too many stems and things like that in there. But I don't think I heard the whole question correctly. You're more than welcome to send me an email. Oberholster@ucdavis.edu.
All right. Well, I think we've just about gotten to 3 o'clock, unless someone has another question. We've enjoyed providing these office hours today. Been fun.
And thank you to Karen for being the guest host on Office Hours with Dave and Anita when Dave and Anita were busy doing something else, I guess.
And don't forget
that
next week we'll be talking about the impact of smoke exposure on grapes and wine. Same link, same time. We'll have Anita Oberholster, Elizabeth Tomasino, and Tom Collins here to talk to you about some of the work they've done and answer some of your questions. So with that, have a great week.
I'm just messaging here. Thank you, everybody. Have a wonderful week. See you next week.
See you next week.
Stop recording. You can stop.
Oh,